There we go. Hello and welcome. Today we are going to be talking about the pathology of respiratory tuberculosis, a bacterial infection of the lungs caused by the inhalation of a pathogen. This tiny bacteria inhabits the apex of the lung as it is an aerobic bacteria, and that location is the highest concentration of oxygen. I chose to research tuberculosis, commonly known as TB, the bacterial disease of the lower respiratory system. This subject is personal to me as my grandmother contracted tuberculosis in 1937 when she was 5 years old. Where she grew up in southern Ohio, the disease was known as consumption and was prevalent due to risk factors such as compromised coal miners' lungs. She was not expected to live. However, she beat the odds and lived to the age of 87 after a 2-year childhood battle and a praying grandmother. According to the Mayo Clinic, tuberculosis is an infectious disease caused by an airborne bacterial pathogen which presents serious detriment to the human body and commonly affects the lungs. Symptoms of TB infection are persistent cough, 3 weeks or longer, coughing up blood or sputum, weakness, fatigue, and pain in the chest. On March 24, 1882, Dr. Robert Koch announced his discovery of the bacterium that causes TB. This is the same doctor known for Koch's postulates, a sequence of experimental steps for directly relating a specific microbe to a specific disease. As you can see here, we can go through these steps quickly. The microorganisms are isolated from a diseased or dead animal. The microbes are grown in a pure culture, as you can see. And then number 3, microorganisms are inoculated into a healthy laboratory animal. And then the disease is reproduced in the laboratory animal. The microorganisms are isolated from the animal and grown in pure culture, and then they're identified. And back in number 2, they were identified firstly. The bacterium that he discovered using his postulates here is known as Mycobacterium tuberculosis, the causative agent of tuberculosis. This human pathogen is slow-growing, non-endospore forming, and rod-shaped. It is aerobic and stains acid-fast. The mycobacteria name is derived from myco, which means fungus-like, referring to a periodically observed filamentous or thread-like growth pattern. And you can see that here in this slide with the little thread-like growth pattern that it has, the little red stained threads there. For pulmonary tuberculosis, transmission of the bacterium is in respiratory droplets. Although tuberculosis can affect other organs, as an example, peritoneal tuberculosis is due to the ingestion of the bacteria, we will focus on the respiratory tuberculosis, disease of the lungs. If an infected individual coughs, the bacterium can be suspended in air for hours. The bacillus can then be inhaled by another person and may cause infection. However, 90% of the time, healthy individual's immune system will defeat the invading pathogen and he or she will be completely unaware of exposure. This additionally is dependent on the infecting dose. In some of my research, I found, and in my personal experience with my grandmother, I found that individuals with compromised immune systems, let's say under excessive stress or other health conditions, let's say HIV, for example, those caused high risk factors for the onset of tuberculosis disease, so that the body wasn't able to fight the pathogen. And interestingly, a mouse model study published in 2018 in November by scientists Alexia Dumas et al. Found that microbiota dysbiosis, an imbalance in a person's microbiota, if you will, resulted in an increased early colonization of the lungs by Mycobacterium tuberculosis during the first week of infection. Armed with this information and the potential for expansion of knowledge in this area, we may find an underlying connection between individuals' specific microbiota and one's ability to resist tuberculosis. Another illustration in the uniqueness of an individual's immune system defenses is a tragic yet enlightening case from history. In Germany, 1926, an accidental inoculation with a TB vaccine generated data regarding variation in resistance to this microbe. 249 babies were accidentally inoculated with a virulent TB instead of the attenuated vaccine. What was discovered is that about 70% of the babies did not become seriously ill. However, 76 children did die. This is known as the Lübeck disaster, named for the location of the event, Lübeck, Germany. In the pathogenicity of the disease, inhaled tubercle bacilli reach the alveoli in the lung even after overcoming several infection barriers. For example, cilia and enzymes in the conducting zone aim to protect the lungs. Alveoli are sacs in the lungs where oxygen and carbon dioxide are exchanged from the blood capillaries. These alveoli are quite prolific and constitute an area of gas exchange equivalent to 70 square meters, or if you're an American, 750 square feet in an average adult. In the alveoli, the bacilli are ingested by macrophages. However, some of the microbes tend to survive. This presents an asymptomatic infection, or otherwise known as latent TB. Unfortunate to the infected human, M. tuberculosis can evade phagocytosis by the alveolar macrophages, which allows the bacteria to multiply inside the phagocyte, commonly ending in death to the phagocyte and release of the pathogen. When tubercle bacilli multiply inside the macrophages, it causes a chemotactic response, and other defensive cells come to assist in the fight against the pathogen. The layering of macrophages and defensive cells gives the first indicator of the name tuberculosis, which is derived from tubercle, which means lump or knob. In the next few weeks, as macrophages die, this forms a caseous center. The bacteria may remain latent in this environment. The disease may be arrested, or the bacilli grow due to the formation of a tuberculous cavity inside the caseous center. Since the bacilli are obligate aerobes, they multiply in the air-filled tuberculous cavity outside of the macrophages. If the disease is arrested, the calcified tubercle will show on X-rays, and it's called Ghon's complexes. Or if the tubercle ruptures, the bacilli are released into the body. And so we're just going to review those slides here. Firstly, you can see the tubercle bacilli that reach the alveoli in the lung are ingested by the macrophages, but some often survive. Infection is present, but no symptoms of disease, so this would be latent TB. Tubercle bacilli multiplying in the macrophages, number 2, causes a chemotactic response, as we discussed, that brings additional macrophages and other defensive cells to the area. These form a surrounding layer and in turn an early tubercle, the knob. It's also called there's a video that you'll see here shortly, and they call it a granuloma. Most of the surrounding macrophages are not successful in destroying bacteria, but release enzymes and cytokines that cause a lung damaging inflammation. So another pathology. Number 3 here, after a few weeks, disease symptoms appear as many of the macrophages die, releasing tubercle bacilli and forming a caseous center in the tubercle. The aerobic tubercle bacilli do not grow well in this location. However, many remain dormant, there you go, latent TB, and serve as a basis for later reactivation of the disease. The disease may be arrested at this stage and the lesions become calcified, the Gones complexes that you can see on x-rays. In some individuals, disease symptoms appear as a mature tubercle is formed right here. The disease progresses as the caseous center enlarges in the process called liquefaction. The caseous center now enlarges and forms an air-filled tuberculous cavity in which the aerobic bacilli multiply outside the macrophages. And number 5 here, liquefaction continues until the tubercle ruptures, which is unfortunate. If that happens, it allows the bacilli to spill into a bronchiole and thus be disseminated throughout the lung and then to the circulatory and lymphatic systems. And you have full-blown TB. Here's a little video to emphasize.
Tuberculosis, also known as TB, is spread through the air from one person to another. When someone with pulmonary TB disease coughs, TB bacteria are expelled into the air in tiny water droplets. These droplets can remain floating in the air for several hours, making it possible for someone nearby to inhale them. Once a person has breathed in droplets that contain TB bacteria, they travel down the trachea and enter the lungs, where they collect in the alveolar sacs. Once in the alveolar sacs, the bacteria begin to multiply. The body's immune system starts working and macrophages begin to surround the bacteria. A granuloma is formed to keep the bacteria from spreading. The TB bacteria remain in the lungs, but the body is protected from disease by the granuloma. In 8 to 10 weeks, the person will most likely test positive for latent TB infection. The progression from latent TB infection to TB disease occurs when the granuloma break open and the TB bacteria multiply. Then the person becomes sick with TB disease and may be infectious. This progression can occur immediately after infection, many years later, or not at all. When the TB escapes from the granuloma and begins destroying a person's lungs, it's called pulmonary TB. TB bacteria can also enter the bloodstream and travel to other parts of the body, causing extrapulmonary TB. Tuberculosis.
Sorry about that. For diagnosis, we have the popular tuberculin skin test uses a purified protein derivative of the bacterium and is injected just under the skin. If there's been a past infection, there is a cell-mediated response from the sensitized T cells and a hypersensitivity reaction occurs. A positive result is a redness around the injection site, which takes a few days to develop. And the skin test, however, may not be indicative of an active case. It may be a reaction due to previous infection or vaccine. Additionally, the diagnosis of active TB from a laboratory can be slow and unreliable. Mycobacterium tuberculosis can take 3 to 6 weeks to form a culture from a smear of sputum. And microscopic evaluation used throughout the last 125 years doesn't always hit the bullseye. Approximately half of the cases examined under microscope are overlooked. In developed nations, new PCR tests, polymerase chain reaction tests, methods are showing promise in the ability to more quickly diagnose the TB infection with greater specificity than the TB skin test. Most exciting is a new automated PCR expert MTB/RIF, which takes an hour and a half to process. This test detects Mycobacterium tuberculosis and tests for resistance to the first-line antibiotic, rifampin, at the same time. Treatment. At the time my grandmother had tuberculosis, she was given not only prayer, but they talked about compresses of onions on her chest. And also she had radiation treatment. She remembers going to Columbus, Ohio, the capital, to have radiation treatment, which was known for bactericidal effects, and it was used for resistant TB at the time. Treatment for tuberculosis with the antibiotic streptomycin began in 1944, and it's still in use today. And I'm not sure, they were not treating her with streptomycin at that time because she had it prior to that. She was diagnosed in 37, so she would not have received that. However, now the short course treatment is quite extensive, lasting a minimum of 6 months, and strict adherence to the therapy to be effective. And as you may recall, my grandmother was treated for 2 years. They didn't have the antibiotic streptomycin at that time, but her treatment was a 2-year-long process. The first-line drugs used in the United States now are a cocktail of isoniazid, ethambutol, pyrazinamide, and rifampin. And there are second-line drugs available as well. Unfortunately, extensively drug-resistant strains of tuberculosis do exist and are essentially fatal. In 1990, it was estimated that 70% of the world's school children received the BCG vaccine. This name is from Bacillus, Calmette, and Guillain. French scientists Calmette and Guillain isolated the bacillus of M. bovis, Mycobacterium bovis, the causative agent of bovine tuberculosis, and made an avirulent version by artificial cultivation over a long period of time. Although the vaccine has been available since the 1920s, we don't use it here in the United States. Its effectiveness seems to be tied to the age at which it's administered. Though considered reasonably effective for young children, it seems to have nearly zero effectiveness for adolescents and adults. Even more tragic is that children who are in the greatest need of the vaccine, for example, HIV-positive children in Africa, they commonly develop a fatal infection from the vaccine. Based on information from the World Health Organization and the Center for Disease Control, tuberculosis disease causes death to about 1.5 million people each year. In most parts of the world, it is still an epidemic. Additionally, due to the compromised immune system of an individual living with HIV, TB disease becomes an AIDS-defining condition. And although treatable in developed nations, TB is the leading cause of death for people who have HIV infection. And in this little infographic, you can see some of the information. An estimated 862,000 people living with HIV worldwide fell ill with TB in 2018. TB is the leading cause of death among people with HIV. HIV and tuberculosis are unfortunately a comorbidity. In history, several key figures contracted tuberculosis. King Tut, the famous Egyptian pharaoh's mummy, reveals that he had the disease. The painter of the 1880s, Paul Gauguin, contemporary of Vincent van Gogh, passed away. He died from tuberculosis. Adolf Hitler committed suicide, but he also had tuberculosis. And fortunately, during the first part of his musical career, the renowned guitarist Carlos Santana recovered from a TB infection after 3 months of treatment. We know more today about tuberculosis than when King Tut had tuberculosis or when my grandmother had tuberculosis. Also, TB treatments have promise in developed nations. However, more work must be done to make responsive treatments affordable and accessible for the world population where the risks of tuberculosis are higher than they are here in the United States, for example. Even when treatments and recovery are achievable, the long-term complications from TB persist. With continued efforts, I am hopeful that tuberculosis can be eradicated. Thank you for watching. Here are my references. And I hope you have a great day.